Hello, AP Biology students. We're going to go over the 12th section in Unit 6. And in this section, we're going to cover something called eukaryotic regulation. Remember, we just got done talking about prokaryotic regulation. That is what we see typically in bacteria. Now we're going to talk about what we see in eukaryotic cells, or cells like our cells. Before I even really begin this section, I'm going to say this is probably one of the most difficult sections in the whole course. The reason is there are so many ways that genes are regulated in eukaryotic cells, and just to group them into 5, you really just don't get the whole story. What my main focus for this section is just to show you some of the ways that cells regulate how much protein is going to be produced in their different ways. There are going to be a lot of minute details that I go over that I don't really want every student to memorize. But generally, students should have a somewhat good understanding of these 5 different types of regulation. I'm also going to break a major rule with my PowerPoints. I put a lot of words and just definitions on one single slide or one single topic here, and the reason is just I want to focus everything that goes over each one of these types of regulation on one slide. With that being said, let's go over the 5 different types of eukaryotic gene regulation. The first type is chromosome structure, and this is just basically regulating how much RNA polymerase has the ability to access the genes. Remember that chromosomes are just made of DNA and protein that can be found in the nucleus of eukaryotic cells. If we regulate how much the RNA polymerase can actually access or bind to the DNA, we can thus regulate how much protein is going to be produced. So what the chromosomes can do is they can either be methylated or acetylated. When methyl groups are added to the chromosomes or the proteins inside the chromosomes, they'll actually compact them a little bit tighter, and what happens is the RNA polymerase can't actually bind to the genes once this happens. Once acetyl groups are tagged on these proteins, it'll actually open up these DNA strands a little bit more, allowing the RNA polymerase to have access to these genes. By compacting or loosening the DNA strands, you will actually regulate how much genes are expressed or proteins produced by just allowing or not allowing RNA polymerase to bind to the promoter. We sort of call this epigenetics. I have this kind of in this section just because we are controlling what genes are going to be turned on or turned off, and thus it kind of deals with epigenetics a little bit. This epigenetics field is a newer field, and we're still figuring out how environments play on or regulate genetics. Our next type of eukaryotic gene regulation is the most crucial because if you stop transcription, you stop a lot of the waste that could build up. So if you stop transcription, you're stopping mRNA from being produced, and if you stop mRNA from being produced, you're stopping proteins from being produced. And this will in turn make the cell much more efficient. What happens is we have these things called transcriptional factors, which are just proteins that will bind to different portions of the DNA. And what they will do is they will either enhance or they will repress the transcription. There are certain locations on the DNA called enhancers that bind with activators, and certain locations called silencers that bind with repressors. And like the name implies, they either activate or repress DNA from being transcribed. And a lot of times what will happen is you'll create this hairpin loop of DNA that will work with these activators and repressors. Next is post-transcriptional control, and again, there's a lot of words here. Basically, after the mRNA is produced, there can be modifications that happen to this mRNA. Typically, we call this actually pre-mRNA. What happens is something called splicing can occur, where we're going to take out certain sections of the mRNA to produce different proteins. The sections in the mRNA that are used are called exons, while the introns are left behind in the nucleus. What this allows the cell to do is use one gene for many different proteins, because essentially you can have a bunch of different these combinations of exons from one gene itself. Next is going to be translational control, and this is going to be the regulation of protein synthesis. So this is after transcription has already occurred and mRNA has been produced. The first way that the cell is going to interfere with this translational control is by using something called RNAi. This is basically where enzymes and RNA will actually interfere with the RNA once it doesn't need that RNA to produce a protein. This RNAi is actually a really new topic within science that we've been finding more and more about. But basically this just interferes with the ability of mRNA to produce a protein. The lifespan of mRNA is also important. Again, it's going to be shorter in prokaryotes just because they have a shorter lifespan and they have to quickly regulate their bodies to their environments, while it's a little bit longer in eukaryotes. But again, basically all we're trying to do here is regulate how much proteins are being produced. If we regulate how much proteins are being produced, the cell can be more efficient at using its resources. Our last type of eukaryotic gene regulation is called post-translational control. This is again after translations occurred or after the proteins have been produced. What can happen is these proteins can be actually modified to fit a specific structure and function that the protein needs, like chaperone proteins helping modify and fold the protein. Or what can also happen is a ubiquitin marker is tagged on a protein that's not needed, that's going to signal it to be broken down by something called a proteasome. A proteasome is basically just a big recycling factor for proteins. What will happen is this proteasome will find tagged proteins and break them down into smaller peptides and maybe even amino acids. Once that happens, the cell can reuse those amino acids to make more proteins. But this is just a way for the cell to control what proteins are actually present in the cell so problems don't arise by having too many of a certain type of unneeded protein. But again, just to end this section, this is a tough section because there's so much information here. But I, as a teacher, would want my students to learn, hey, what's the main concept of every one of these different types of eukaryotic regulation?